Superstar.
Appreciate that she took the afternoon off the track to come and work with us. So she spent last summer working in my lab with her partner Eli, looking at the kinetics of the gamma-2 ADH. She took it. This is nice of her to do biochemistry for us because I know that her long-term interest is in doing graduate work on the brain. So this is her first part, but I think there's some connections that she's made. So it was my privilege to have her, and I'm looking forward to see what she does in the future.
Thank you, Dr. Chase. Good afternoon, everybody. So I had the privilege of doing research with the enzyme gamma-2 ADH. For this presentation, I'll be speaking to you firstly a little about retinoids and how this affects everyone in the room and also how it applies to my research. Then I would give you a brief overview of some of the procedures that we carried out, like the enzyme purification and the kinetic assays, and the results that we got from the kinetic assays, the kinetic constants, and how we determine what mechanism our enzyme binds by, and also how it affects you in general, like I said before. So firstly, retinoids are a class of vitamin A or retinol, and this is taken into the body as retinol esters, which can be found in fruits and vegetables, mostly the orangish, yellowish colored vegetables, like carrots, and also liver of animals. Once this is taken into our body and digested, it is transported in the blood to the liver where it is stored and to other cells in our bodies. What we're most interested in is what happens to retinol when it reaches the cells. Retinol is oxidized by alcohol dehydrogenase in the cells, and then further to retinol, then further oxidized by retinaldehyde to retinoic acid. Retinoic acid is important to us in transcription activation. Retinoic acid binds to a retinoic acid receptor, which then binds to the retinoic acid response element, which allows for transcription activation, which leads to targeting genes, cell growth, differentiation, and apoptosis. As I said earlier, retinol is oxidized by alcohol dehydrogenase to retinoic acid. However, this oxidation can be inhibited in the presence of alcohol, and therefore we have a low production of retinoic acid, which hinders the cycle that I showed previously, preventing any of these from occurring, which can lead to problems in the cell. Therefore, for our overall hypothesis over the summer, which was first set forth by Kulikov and Duster in 1991, it states that a high alcohol intake can lead to inhibition of retinol oxidation by alcohol dehydrogenase, which leads to decreased retinoic acid production, leading to dysregulation of growth, apoptosis, and differentiation, which can cause cancers, fetal alcohol syndrome, and cirrhosis of the liver. Our main focus over the summer was quantitatively how much does alcohol inhibit retinol oxidation and how much does it hinder the rate of oxidation of retinol by alcohol dehydrogenase in the presence of alcohol. Alcohol dehydrogenase, as one of my colleagues, Kat also did research on the same enzyme as I did. It's an isoenzyme and has different forms. My group focused on alcohol dehydrogenase 1C2, which is a different form to what Kat and her partner focused on over the summer. Alcohol dehydrogenase 1C2 can be found in the liver, the stomach, and small and large intestines. This also exhibits two polymorphisms, alcohol dehydrogenase C1 and C2. We dealt with C2 specifically. C1 is known to be found in East Asians and Africans, and C2 is known to not be ethnospecific, meaning it's distributed evenly among different ethnicities. Previous research has stated that alcohol dehydrogenase C1 has a greater susceptibility to alcoholism and cancer than alcohol dehydrogenase C2, which is the gene that my group looked at over the summer. However, we don't know how this affects it quantitatively, how it acts on alcohol how alcohol acts on alcohol dehydrogenase C2 quantitatively. So what we want to look at is calculating the rate of retinol oxidation using this big equation right here. With this equation, we will insert the kinetic constants that we found over the summer and also the concentrations of our substrates, and it will give us a value for retinol oxidation. This equation also allows us to assume the order of binding for our enzyme, whether it's in an ordered fashion or whether it's a random fashion. Previous literature did have some kinetic constants that we could use to put into the equation, but as you can see, there are many blank spaces, especially at 37 degrees Celsius, which is important to us to understand how the enzyme acts at physiological conditions, which is in our bodies. This is where we come in. And for our summer, we wanted to determine the kinetic constants of gamma-2 ADH specifically, since so little is known, at both 25 and 37 degrees Celsius. We also wanted to determine the kinetic mechanism by which this enzyme binds. We were given a plasmid which expressed our gene and transformed by Steve Pekovich, one of our lab personnel. He did this for us, thanks to him, he's not here right now. And we then were able to produce our enzyme by growing it in a media of E. coli and inducing it with IPTG. IPTG is a lac outgrown which increases production of the enzyme and also inducing it with zinc sulfate, which can be found in the activation site of the enzyme. Once we grew a sufficient amount of the E. coli with our gene expressed in it, we then lysed the cell in order to break open and set free the proteins that were in the cell. With the proteins that we acquired from the lysing, we carried out a number of steps of purification. And after each step of purification, we collected a sample whereby we can run a SDS polyacrylamide gel electrophoresis to observe the level of purification that occurred at each step. These earlier steps, we can see that there are a number of bands in the lanes, which shows that we have proteins, unwanted proteins present in the solution still. So we continue purification until our elution step, where we can see only one band that is present at 40 kilodaltons. This band helps us to confirm that we did indeed purify to achieve gamma-2 ADH enzyme, and we're now able to move on to our step of kinetic assays and able to determine kinetic constants for our substrates. Our kinetic assays were done in a buffer of 50 millimolar sodium phosphate and 250 micromolar of EDTA. For each assay, we had ethanol and NAD with the buffer along with some of the enzyme, and then retinol and NAD with the buffer with some of the enzyme. We also varied the concentration of NAD and ethanol and varied the concentration of NAD with retinol. We allowed these to react for 1 minute in the spectrophotometer at both 25 and 37 degrees Celsius at a rate of 340 or 400. These rates were fit to sigma plot. For our results, we were able to achieve a saturation curve. As I stated earlier, we varied the concentration of our NAD, and we also varied the concentration of the ethanol. This is basically what this graph is showing. For each point here, we did these tests twice for each concentration, probably more times, but this is one of our most perfect data that we received actually over the summer after running all these assays. This is a graph representing the most perfect data that we had for ethanol at 25 degrees Celsius. And we were able to take these rates and plug it into a computer program known as sigma plot, which allows us to fit it to the best fit line. And it also tells us what order did our substrate bind. For the ethanol and NAD, we found that it's bound by, ordered by mechanism. We also have results for this for retinol and NAD at 25 and 37, and ethanol at 37, but this is just a sample of some of the results that we received. Also here we have an R-squared of 0.982. What this R-squared tells us, it's so high that we have enough confidence in the kinetic constants and the values that we produced from our data. With the values that we got, we were able to add it to the unknowns that I showed you earlier that were missing in literature values that had been carried out previously. Although, however, there are some discrepancies in the values that were found in the literature and what we observed in our study over the summer. This can be explained because for the literature values as we read them, they did not vary their NAD concentrations as we did, receiving 5 saturation curves. They only had 1 saturation curve. As I showed you earlier, we had 5, but in the literature they only had 1 saturation curve. So this allows us to accept our data with more confidence because we had it to measure against 5 instead of just 1. Also, most importantly, we found that there is indeed a difference, an effect of the temperature on our kinetic constants. As you can see down here for the KIA of NAD with retinol, the substantial difference between the two temperatures, and we were able to know that 37 degrees is important to us when performing kinetic enzyme assays. So for our future work, what we hope to do is use the kinetic constant that we got for retinol and ethanol in order to help us predict inhibition of ethanol and retinol, but but we also need the kinetic constants of the reverse reaction of this enzyme, because what we did over the summer was only the forward reaction. We also need to know what happens in the reverse reaction. Therefore, we need to know the kinetic assays for NADH, acetaldehyde, and retinaldehyde. Once we have these kinetic constants, we can then take each value and plug it into the crazy equation I showed you earlier for velocity, which can help us determine the rate of retinol oxidation. So for the summer research, after all the hard work that we did, my partner and myself did, we were able to get substantial kinetic constants to add to literature value, and hopefully we will be able to add to that literature value that has been presented previously. We have not written ours up yet, as Dr. Chase was telling me, but hopefully we can add to it, and they will have more information to know what really happens at the molecular level with gamma-to-gamma-to ADH. We also were able to determine the mechanism using sigma plot, and we found that ethanol and NAD bind in ordered bye-bye, but retinol and NAD bind in a random bye-bye. We also found that temperature matters. I've probably said that a million times, but it does matter that you do enzyme kinetics. If you're looking at physiological conditions, you want to do it at 37 degrees Celsius. So with the few data that I have, we're not able just yet to know what number and what rate is retinol oxidized, but we do have preliminary knowledge that tells us ethanol does inhibit retinol to some extent. In our research, we found that the km of ethanol is 1 millimolar, and it is known that legally drawn can be approximately 20 millimolar in our system. This is 20 times, legally drawn is 20 times more than what we found to saturate the enzyme, which clearly tells us that gamma-to-gamma-to ADH is highly saturated by ethanol in our system. We also found that the km of retinol is 20 micromolar. However, in vivo, in our cells, it is approximately less than 1 micromolar. This is 1/20th. This is less than what we have up here, leaving the enzyme unsaturated. So we therefore have a substantial inhibition of processing vitamin A or retinol towards retinoic acid, which can lead to problems in the cell, like I stated in our hypothesis. So I hope after this talk and bearing with my accent that you were able to understand the hypothesis that was set forth by Purlock-Induced and how we used it to apply to our research over the summer, and how ethanol affects retinoic acid and also leads to diseases. And we've also seen our contribution to the literature and the amount of kinetic constants that we had to add to the literature over the summer. I would like to thank these people for, well, firstly I'd like to thank Dr. Jennifer Chase for the opportunity to do research with her, and Embry for the grant in order to continue research, and Dr. Boschman and Hurley, who gave us the plasmids with the gene, and Steve and Nancy, who were great help in the lab over the summer. Any questions?
No.
I'm too shy.
I just know all the answers already.
Oh, I was gonna leave. Oh, I gotta go.
What? What should we actually do? It would be very useful. So I just have a bunch of random sentences. None of them.
Whatever I can manage to catch.
And like I connect them, but I know that if you really knew what you were talking about, none of them would make sense. I'm just going off the track.
Let's go.
Like, and it's all there, so the effort was put forth. That's what I'm feeling.
The effort's totally there. Nice.
My parents, yeah. I was doing that in front of my uncle.
I feel bad. Like, were we really supposed to be able to ask questions over this stuff? Oh, you like that?